Glycolysis. Primarily I'll be referring to what's required for the OCR A2 specification in biology. If you're using that specification and you have the recommended textbook, you need page 138, figure 1, which will help you understand the process of glycolysis. Now, glycolysis is the first step of respiration, and it does not involve the mitochondrion. So we are looking at the location of glycolysis, which is in fact the cytoplasm. Now, the actual stages of glycolysis, looking at this diagram, seem pretty complex. There's 10 enzyme involved reactions taking place. There's a change from glucose to another sugar, fructose, and at some point it splits into two to get several products here. This looks a little bit complex. We need to simplify this, and your textbook simplifies it greatly, but I just need you to understand what's going on in the main steps. So let's break it down. The actual respiratory substrate in glycolysis is glucose. So glucose has just entered the cell, and one of the first steps that happens is that it needs to become phosphorylated. Now, the source of phosphates here is a molecule of ATP. ATP you should have already studied. ATP will phosphorylate the glucose, so it releases a phosphate, and that phosphate sticks onto the glucose molecule, and ADP is released, and we get what's called glucose 6-phosphate. The 6 because the phosphate is stuck onto carbon number 6. This happens again. So we get ATP, releases its phosphate to make what we call hexose bisphosphate. Look at the letters Bi there, meaning 2 phosphates. Now we've got one on carbon 6 and on carbon number 1. Look at the change in structure. We've now got fructose. You don't need to know this for OCR biology. Just be aware that glucose is phosphorylated twice to make hexose bisphosphate. This molecule now splits up. It splits up into 2 triose phosphates. Why triose? Because both of them have got 3 carbons. If you look at the main diagram, the complex diagram again, there's dihydroxyacetone phosphate and there's glyceraldehyde 3-phosphate. They're the 2 triose phosphates. The former dihydroxyacetone phosphate converts to glyceraldehyde 3-phosphate. It's too complex for OCR biology. We don't need that. You just need to know there's 2 triose phosphates here. Now we get to the business end of glycolysis now. The 2 triose phosphates are what we call oxidized. In other words, they're going to lose hydrogen atoms. So we need a reducing agent for that. And what gets reduced is this coenzyme called NAD. Nicotinamide adenine dinucleotide. And this is going to take off the hydrogen atoms and become reduced. And this gives us 2 molecules. 2 molecules of reduced NAD because we had 2 triose phosphates. Along this pathway, we also now need to take the phosphates off. So we need to dephosphorylate the triose phosphates, and this time phosphorylate ADP. But to begin with, we need to add some more phosphates in. So at the same time as reduction, we also get 2 phosphates from the cytoplasm joining to the triose phosphates. Otherwise, our numbers don't seem to make sense at the end. You'll see why. So in comes 4 molecules of ADP, phosphorylated. The phosphates are now removed from the triose phosphates. So let's recall what's happened so far. At the start of this pathway, we used up 2 ATPs to make glucose 6-phosphate and hexose bisphosphate. We're 2 ATPs short. We're trying to make ATP, but we're 2 down already. But from triose phosphate, we've just generated 4 ATPs. So that gives us a net gain now of 2 ATPs. We've actually made some ATP. We've transferred some energy from glucose into ATP. So we have dephosphorylated triose phosphates and we have oxidized the triose phosphates. That leaves us 2 molecules now of pyruvate. That's glycolysis. That's the products of glycolysis. Okay, back to our main diagram, our very complex diagram. Let's break it down. This shouldn't be so difficult now, although you don't need to know such detail for OCR. It does help you now, I think, to have a good understanding if we follow this diagram. There's the first bit. Glucose is phosphorylated using ATP. There's the second bit. Glucose is phosphorylated again. And now we've got fructose 1,6-biphosphate, or in other words, our hexose bisphosphate. That molecule then splits up into triose phosphates, 2 of them. Then we get our oxidation and reduction of NAD. Now on this diagram it only seems to happen once. That's because it's not showing what happens to both triose phosphates, it's only showing what happens to one of them. So this happens twice, and therefore 2 reduced NADs. There's our phosphorylation of ATP. And there's our second phosphorylation and our production of pyruvate. Don't forget this happens twice. So net gain of 2 ATPs, 2 pyruvates, and 2 NADs. One more key term for you. You might have heard of oxidative phosphorylation when you've studied electron transport chain in photosynthesis. We'll be doing that again with respiration. But this type of phosphorylation is called substrate level phosphorylation. So there's a key word for you to be able to use.